When you order a plasmid, such as from Agene, one of the most important places to look is the bacterial resistance. The bacterial resistance is going to correspond to the antibiotic resistance that the plasmid has. But sometimes you might actually see that there are multiple antibiotics listed, and only one of those antibiotics is going to actually correspond to the antibiotic resistance gene that is on the plasmid that you're ordering. You might be confused about this, but it's actually that the second antibiotic is needed because of the type of cells that the plasmid is coming to you in. A common example of this are things like RIL strains of BL21. In these cells, the bacteria have an extra plasmid in them already before that plasmid that you ordered was actually put in there. And this extra plasmid has the instructions for making extra copies of rare tRNAs. And so the tRNAs are going to be the RNAs that actually bring the amino acids to be added to the growing chain when making a protein. And it turns out that different types of organisms use different codons in different proportions. And so the genetic code is degenerate, so there are multiple codons that code for the same amino acid. And so it's kind of like how you can spell gray with an A or gray with an E. And if you were to be in the United States, you would see it more with an A. And if you were to go into Europe and England and things, you might see it more with an E. Similarly, different organisms will use some codons more than other codons. And so we can get bacteria to make proteins from any organism, but if those organisms were using different spellings of the same words, then what would happen is that the bacteria might be kind of slow at making our proteins. And one of the strategies that you can use to get around this is by giving the bacteria more copies of the tRNAs genes that are going to correspond to the codons that are found in the gene that you're putting in with your plasmid, rather than what that bacteria naturally has. And this is the strategy taken by some types of cells like RAPL, RAL, RP, Rosetta cells. And so for RIL, the RIL, this is saying arginine, isoleucine, leucine. It has extra rarer codons for these. And this is good for organisms with AT-rich genomes that use those codons more. This is an alternative strategy to codon optimization, where you actually go and you go through and change the codons that are rare to ones that are more frequently found in the E. coli. In this strategy, instead, you change the E. coli themselves. So they have more copies of those tRNAs. So if you think about the gray versus gray, maybe if you're thinking about one of those word magnets, the magnetic words that you can make poems on and stuff on your fridge, it's like ordering more of the gray with the alternative spelling. So you have more of those, and therefore you can make more sentences that are familiar in the particular context. And so in this case, you have more tRNAs of those rare codons so that the bacteria doesn't get stalled, because the bacteria is going to stock up on the ones that it wants for its proteins, not the ones that you want for your proteins. So you can give it more of the ones that you need for it to make your protein. And this is the strategy for these cells. Because these tRNA genes are on a separate plasmid, it's great in that you can put lots of different plasmids into these cells, and all these cells will always have that plasmid. But it's not so great in that you have to then add a second antibiotic in order to make sure that the cells are keeping both that plasmid and the plasmid that you're putting in there. Because the plasmid that you're putting in there has an antibiotic resistance gene, so that only the cells that have the plasmid will be able to grow in the presence of that antibiotic resistance gene. But if you don't add that second antibiotic, well, now you're not going to have the selection for that second plasmid. And so you need those two antibiotics, one for this extra plasmid and one for the plasmid that you're putting in there. And so the chloramphenicol, if you're looking at an RAL strain, the chloramphenicol is going to be corresponding to that plasmid. And one other practical note, chloramphenicol is not water soluble, and so you're going to want to make a stock of it in ethanol. For some, like with RIPL, here they're adding, you actually have two plasmids inside of there along with your plasmid. And so you have to have three antibiotics. You got to have the chloramphenicol, the strep, and the antibiotic for the plasmid that you want. But sometimes that's what you got to do in order to get your proteins made. And these are often going to be in the context of BL21 DE3 cells, which are a common strain for expressing proteins. And so this DE3 part makes it so that they're able to actually make protein on demand. If you use a T7 polymerase system, the DE3 part is going to make it so that you can make the T7 polymerase on demand. And then if you have a T7 promoter in front of your gene, then you can make your protein on demand. These cells are great for making protein, but not so great for keeping the sequence long term. And so for like cloning, if you're just trying to amplify the plasmid rather than trying to make protein, you often use cloning cells like DH5 alpha, XO1 blue, top 10, and these don't need to have that extra codons because you're not caring about the codons. You're not caring about the proteins that are being made. You're caring about the DNA being made. And so, for example, when we have this cloning plasmid that we've ordered, and so this is basically we're ordering a plasmid with a different tag so we can clone our gene into this plasmid. So we just care about the plasmid itself rather than protein made from the plasmid. And we see that this one is found in this DH5 alpha cell. So it's coming to us in these cloning cells. Whereas this one, so this is the plasmid for TEV protease, and so we actually want this protein, which is going to allow us to cut the tag off of the protein that we eventually make with this plasmid. In this case, this is coming to us in those BL21 DE3 RIL cells. And so it's in those expression cells that have that extra plasmid with the rare codons. And so you don't always need this. And so most of the time I just use straight BL21 DE3. But for some proteins, they express better when they're in these strains with those tRNAs for those rarer codons. So with this strain, what I'm going to do is we're going to grow it up and we're going to purify the plasmid, and then we're going to clone our gene into this plasmid. And then once we grow up a lot of it, we're then going to purify it and we're going to check the sequence and everything, and then we'll stick it into the expression cells and have protein made. But in this case, we don't need to purify out the plasmid. We just need to grow up more of these cells, and then we can use those cells to make the protein for us. Although it's still a good idea to purify the plasmid, stick it in some cloning cells, and then you have it for longer term. But you can make glycerol stocks from this versions in the cells already, and this way you don't have to pay for more of the expression cells. But you do need to remember that you're going to need that second antibiotic in order to make sure that you're keeping the second plasmid in those cells. So the first antibiotic is from the plasmid that you're caring about, that you think you're ordering, that you hopefully are ordering. And the second antibiotic is for the cells that the plasmid is in. More specifically, to make sure that the cells keep that plasmid that has the instructions for making those rare tRNAs, that recognize those rare codons, so that even if that's not the one that the bacteria typically uses, it'll now have plenty of those tRNAs in stock so that you don't get stalled up. An alternative strategy is to go and actually get this gene codon optimized. And so this might make sense if you're ordering the gene itself from some sort of company, that then you can do the codon optimization when you're ordering it. But if you're cloning it out of somewhere else or that sort of thing, then it might be easier to just go ahead and use different strains of E. coli, although they might charge you a little more. But if you're ordering a plasmid already, sometimes that plasmid will come in those cells. And so hope this helped you understand why you might have those kind of multiple antibiotics that you're needing to add.